Good morning all of you. Today's class is color vision. Objectives of this class will be to know about the physics of color, 2. the theories of color vision, 3. to know about the neurophysiology of color perception. What is color sense? Color sense is the ability of the eye to discriminate between the colors excited by the light of different wavelengths. So the color is nothing else but the wavelength is what we are recognizing. So when we took a beam of light, white light, and passed through a prism, we got the rainbow colors, 7 different colors because of the different wavelength. So this colors which we are able to see is called as visible spectrum. Human eye is sensitive to the light rays in visible spectrum. These are nothing else but electromagnetic waves. They give the colors, spectral colors, which is VIBGR, violet, indigo, blue, green, yellow, orange and red. They have the wavelength between 400 to 750 nanometers. Less than 400 is ultraviolet rays, more than 750 is infrared. So human beings have an ability to see more than 100 different colors. When you look into this picture, you can see this is a narrow spectrum which is visible to us, and the wavelengths of larger wavelengths they are infrared and they are not visible to our eye, and the smaller wavelengths like UV rays, gamma rays, microwaves, all these things are not seen by us, cannot be seen by us. So what are the attributes of color you must know? The first one is a hue. Hue means the pure color. Pure spectrum colors commonly referred to as color names: red, orange, blue, the pure color. The intensity means the brightness of the color, that when you mix with a black, it gives a degree of brightness. Saturation means it is the color when it is giving a pastel color, when it mixed with white, that is your saturation. So again you can see the hue here, less saturation here, more brightness here. Hue is a pure color. Saturation is with your darkness, brightness is with your light, white. So what are the types of vision? Achromatic vision, sensation of white light with no color perception, that is called as achromatic. There is no color vision. Second one is a chromatic vision, where the spectral color vision is there. Extra spectral color vision we can get by mixing of two spectrum. So here you can see the achromatic system, where only brightness is visualized, not the colors. In the chromatic system you have the red, green, blue, yellow, blue systems. What is color vision? Color vision is a function of cones and thus better appreciated in photopic vision. So cones are the ones which are the receptors which are present in the retina, most concentrated in the macula of the retina, and this is very important for the color vision. So cones are the receptors for color vision. As you see here, you have the 3 colored cones here, isn't it? So red cone is there, green cone is there, blue cone. These are the 3 primary types of cones which are present in the retina for color perception. So 3 different types of cones for color vision: red sensitive, green sensitive, blue sensitive cones. You can see here the wavelengths of the blue cone. You can see the rods which is having a higher wavelength, but it is not for the color vision. You have the green cone as well as the red cone. So types of color vision. So when you look into the types of color vision, we are all trichromats. We can visualize 3 colors. So here 3 colors we can visualize. Some apes and old world monkeys also can do that. Trichromats have only 2 colors. They are blue sensitive, red sensitive, and some mammals also have that. Monochromats are marine animals, and tetrachromats are there. Infrared vision also can be seen by your the snakes have it, and they can see the infrared waves too. So gene and color vision. So what is this? Rhodopsin is very important and it is located on the chromosome 3, but this is not for the color vision. Second one is the blue sensitive cone, it is located on chromosome 7, and red and green sensitive cones which are present on the X chromosome. So this is very important. You must remember X chromosome, which is very important for the red and green sensitive cones. This is the most common defect we see in color blindness. So what are the characteristics of the color we must know? One is what are primary colors, two what are complementary colors, what is the effect of illumination, what is Purkinje shift phenomena, what is the effect of background color and the effect of intensity. So primary color is any color when mixed in equal proportions gives some other color. These are called as primary colors. What is a complementary color is any primary color when mixed properly give a complementary color and it gives a sensation of white. So each color has a complementary color like a purple to green, orange to blue, yellow to dark green. You can see here yellow to purple, orange to blue violet, green to red, like that. These are called as complementary colors. So effect of illumination. So what is the effect of illumination? When you see in the daylight, the picture is, you can see a picture here which is different, and when you see in a night light, the same picture is viewed in a different way. So the in dim vision or when the rods are most sensitive, blue color and green color dominates. But in the high illumination during the daytime, the red and the yellow predominates. So there is a shift of sensitivity we know with respect to the receptors. So the images which we view change color depending upon this. This is the effect of illumination. So this change and shift of the brightness of the spectrum, this is called as Purkinje shift phenomena. Shift to the left occurs during photopic vision, shift to the right occurs during photopic vision. So you can see here color termination in the dark, it comes to the blue range. During day it goes towards the red range. So there is a shift during the day and to the right, and there is a shift to the left during the night. This shift because of the illumination is known as Purkinje shift phenomena. So what is the effect of background color? So the background color determines how clearly we can see the other color which is on that. So you can see here in this, the dark background gives a little more prominency for this yellow color. So that is an effect of background color too. So effect of intensity. For the same wavelength of light, when the intensity of the color is increased or decreased, you have different shades. So you can see here the different shades of the same color. So now we will go to theories of color vision. So the theories which we are going to see is trichromatic theory. This is also called as Young-Helmholtz theory. It is also called as pigment theory, retinal theory. Second one is opponent color theory or color theory of hearing. Third one is called as Granit's dominator and modulator theory. Fourth one is Quill's duplicity theory, and fifth one is Retinex theory. So what is the history? This is long back from 4th century. The people were working on the colors, and so many informations about the color perception is given by the start of Aristotle to James Clerk Maxwell and so many other scientists too. So first one is trichromatic theory. This is given by Thomas Young in 1802 and Helmholtz in 1852. So it is named after them too. What this theory says, there are 3 kinds of cones for each primary color: red, blue and green. They have different photopigments, maximally sensitive to one of the 3 primary colors. Combination makes millions of colors. So it does not explain after images or color blindness. So here you can see the picture of Thomas Young and Helmholtz. So trichromatic theory says there is a blue cone which is having a smaller or shorter wavelength, and a medium cone which is a green cone which is having medium wavelength, and a large cone which is having a red color which is a larger wavelength. So the specific cones determines the perception of color. So 3 cone system, here you can see the large wavelength, medium wavelength and small wavelength. So large is red sensitive, erythrolab, M is green sensitive, medium wavelength, and S is blue sensitive and short wavelength. As you see here, you can see there is an overlap of these curves, isn't it? So it not only recognizes the peak of the blue wave, it also blue wavelength, it also recognizes in the range of wavelength this pigment, this cone will become sensitive. Likewise for the green wavelength too, as well as for the red wavelength also. So fovea and colors. What is fovea? which is present in the retina, isn't it? In the central part of the retina, it mediates color vision, contains densely packed cones. Only M and L cones are present here, medium and long length. Fine details of the colors you can see. So here you can see the fovea. When the light falls on the center of it, lot of concentration of the cones are present here, and mostly the red cones and the green cones. Here also you can see the concentration of red and green cones in the center of the fovea. In the peripheries what we have is your rods. So central vision is mostly very important for your color vision. In the visual cortex, how do we perceive a color? So it compares the relative frequencies of action potentials, and it finds out at least two types of cones are required for the color vision. So in this diagram you can see the cones as it perceives the color. The modulation is like a blue, green and red. When it goes to the lateral geniculate body, much more detailed spectrum occurs. Finally, when go to the cortex, we are able to perceive the color as it is. So that was the trichromatic theory. So as it could not explain certain features which we would see normally, there came the opponent color theory or color theory of Hering. So here we have the picture of Eval Hering. What he said is, there are 3 cones, but the response is for 4 colors, 4 opponent colors. He said opponent means opposite to one another. So this is actually an extension of trichromatic theory. 4 primary colors. There is no greenish red or bluish yellow color as we can imagine. We don't see something like that. So red and green color oppose each other, and blue and yellow colors are opposite. Complementary colors becomes opposite to the primary color. So when we look here into this picture, green is opposite to the red, blue is opposite to the yellow, white is opposite to the black. So here you can see the opponent colors, blue against yellow, green against red. So you have the blue cone, green cone and the red cone, and what happens is they oppose each other. When green and blue oppose each other, and red and green oppose each other, and blue and yellow oppose each other. So these sensory receptors comes in pairs, but actually we don't have an yellow cone, but the information from 2 other cones like your blue and green and red gives the yellow color. So you have the opponent cells. So the ganglion cells of the lateral geniculate body and neurons of the visual cortex have this opponent property. Types of color opponent ganglion cells we'll see: red green opponent color cells and blue yellow opponent color cells. So here you can see this blue and green. You can see blue and yellow. Single opponent property. The ganglion cells and the cells of the lateral geniculate body are single opponent. That means what? You have an on-center off-surround cells and off-center on-surround cells. Excited on-center cells. We'll see on-center off-surround cells. Excited when a red stimulus is present in the center of the receptive field, and inhibited when the green stimulus is present in the periphery of the receptive field. What do you mean by this? So here you can see an on-center cell. Okay, these two are the on-center cells. If it is stimulated in the center, in the visual field, in the receptive field, it is excited. If it is stimulated in the periphery by green, it is inhibited. Likewise, you have off-center cells too, wherein which if it is stimulated in the periphery by the red color, it is stimulatory. If it is stimulated by the green center, in the center, it is stimulatory. So likewise, you have for the on-center and off-center for the green colors too. So for the off-center and surround cells. So double opponent property. So what is double opponent property? What happens here is you have the on-center cells here as well as off-center cells here. In the on-center cells will get stimulated if it is stimulated in the center by the red, and it is stimulated by the green in the periphery, then it becomes excited. The same cell, when it is stimulated by the green in the center and by red in the surrounding it becomes inhibitory. Likewise, opposite occurs for the off-center cells. This is very important because this is what gives the second line of image processing and understanding in the perception of color at the ganglion cell level. So here you can see the excitatory neurons. These are the cones, and here you have this your visual receptive fields, and here what we have is your bipolar cells which are excited and inhibited. Here also you can see the inhibitions. Here is the cells, receptor cells, then the bipolar cells and the cones and the ganglion cells. So Herding's opponent theory says there is breakdown and resynthesis of the photopigments. This is what is happening during the color vision. Here we want to understand a concept called after image. This is supported by negative after imaging. You look into this picture, then suddenly you look into a background and you see the opposite colors. So now we'll see this example. You concentrate on this picture in the center, in that white dot for 30 seconds. Okay, so you can do that 30 seconds without changing your, without blinking, without changing your field. Just look into it, stare into it. Then you see this background, white background. What happens? What are you seeing? Are you not seeing the opposite color? Instead of that yellow, you're seeing a blue. Instead of green, you're seeing a red. And instead of black, you're seeing a white. So how is this possible? So here also, we'll explain that. Here also you see, you concentrate on these dots, center for quite some time, for about 30 seconds. Then you again blink your eyes, you can see the green images. This is because of the after image. Why is this happening? So the cause for this is whenever you see a blue color, an yellow color, what happens is in the ganglion cells, all the yellow part is habituated. It is getting weared out. It becomes tired. It goes off. But what happens is the blue part is active. So suddenly when you look into this white background, what happens is the red part of the cone starts discharging. So you give a red vision because this is a spectrum. This white is a spectrum. It has all the colors. But already yellow is weared off. So you can see the blue. As well as like green is weared off, and so you see the opposite color red. Black is weared off, so you can see an opposite color white. So this is called as after image. So here you have two things, negative after image and positive after image. What is negative after image? You can see in this picture, you just concentrate on the dot for some time and then look into this white background. What do you see? You see the opposite colors, complementary colors, isn't it? This is called as negative after image. What is positive after image? Again you look into this picture and concentrate on the black background. Again you see the colors appearing, the colors, real colors appearing. This is called as positive after image. That's it about the opponent theory. Now we go to the Granit's dominator and modulator theory. What Granit did is he introduced microelectrodes into the ganglion cells and investigated the sensitivity of the light to various wavelengths. He found out there are dominators and modulators ganglion cells which are responsible for the color vision. So here you have the picture of the Granit, Ragnar author Granit, who explained well about this theory. So what is a dominator? These are white ganglion cells which respond to the whole visual spectrum but detects intensity of the light but not the color. What does modulators? These are X ganglion cells. These respond maximum to a narrow wavelength of light. There are three modulators: blue wavelength, green wavelength, red, yellow wavelength modulators. So here the blue, yellow, green, red, these are the modulators. These are the X ganglion cells which are responsible for the color vision. So I just give an idea about what is Y ganglion cell. It is the largest cell, and W cells is also there, which is small one. The medium size is the X ganglion cells. The fourth theory is Schools duplicity theory. It was given in 1866 by Max Schools. The duplicity theory of vision concerns the comparison of both the differences and similarities and interaction between cones and the rod system in visual pathways. Rods are responsible for the scotopic nocturnal achromatic vision, cones of photopic diurnal chromatic vision, daylight vision. So the interaction between these two causes the vision. This is the theory by Schools duplicity theory. The fifth one is the Retinex theory, where it talks about the color constancy. So the Lat effect refers to the capacity of the full color image slowly by looking into a photo with the red and the grey wavelengths. You can see this picture here, over here. So the colors in the cars appear different because of the background color. So it shows about certain colors are constant. This is the theory given by the Retinex theory. So now we looked into the theories of color vision. Now we will look into the neurophysiology of color vision, or the mechanism of color vision. What do you mean by that? The process of color analysis begins in the retina and not entirely is a function of the brain. Perception of color has a combined role in the retina, lateral geniculate body and the visual cortex. So transmission and encoding of color signals. When color strikes the retina, it produces a local action potential. Bipolar cells are stimulated, it activates the ganglion cells, it processing in the cones occur here. So the cones which are responsible are the LM cones and the S cones, the Y ganglion, and goes to the Y ganglion, goes to the lateral geniculate body, and it releases in the parvocellular pathway. White and black release in the magnocellular pathway. After processing, the primary visual cortex goes to the primary visual cortex area 4C. Layer 2 and 3 clusters of neurons called blobs are there. So these ones are getting the information from the area 4C and they process the color information. Later on they project to the area V8, which is the association area present in the lingual and fusiform gyri of the occipital cortex, where the color is converted into the sensation of color. This is the neurophysiology or transduction of color perception. Thank you.